Hello, welcome to week 5. This week we're going to talk about chapter 10. In this chapter we're going to focus on DNA structure, the history, how it was discovered, and various ways that you can use to analyze DNA. So first of all, before they realized DNA was the genetic material, they did know that it must have several different characteristics. In order to be able to serve as the genetic material, it must be able to, first of all, it must be able to replicate so that it can be passed from generation to generation. It must store information that those organisms can use. It must be able to express information so that they may have this information that can actually use it. And then last but certainly not least, they knew that it must allow variation by mutation. And this way, different generations mostly are going to be similar, but every so often we're going to get some variation in there. And that's what's going to be the raw material for a new species to form. So one of the first to look at how DNA was acting as genetic material was Griffith, and he performed some transformation experiments. He took mice and he injected them with 2 different strains of pneumococcus, it's a bacteria. There was one of the strains was encapsulated, and he called it the S strain. The reason they call it the S strain is because it literally looked kind of shiny if you were looking at these colonies on a plate. The other strain was a non-encapsulated, or R strain. These looked almost rough. That's where the R designation comes from. If you were to look at them, rough or dull colonies, if you were to look at them on a petri plate. If he injected the S strain, this is the virulent strain, the mice would die. The R strain, not virulent. These mice would live if they were injected. If you heat killed the virulent S strain, so if you remember back from Biology 111, we talked about things like enzymes. If you heat them up, they become denatured. So many of the important enzymes in these bacteria became denatured and inactive. He injected these into these mice and they still lived. However, that genetic material is still there. When he combined these heat-killed strain with the R strain, what he expected was that, well, this one doesn't kill mice, this one doesn't kill mice. You put them together, it shouldn't kill mice. But sure enough, it actually did. And when he took samples from these mice, from this dead mouse, he found that it actually contained the live virulent S strain. And so what happened, we call this process transformation, but the genetic material from the S strain, although the bacteria itself was dead, the genetic material was still there. The non-virulent strain was able to take up that bacteria and become virulent. And because this bacteria was alive, it was able to replicate in the mice and cause severe illness. So another incredibly important discovery was research that was done by Avery, McLeod, and McCarty. They took Griffith's experiments in transformation, they took them one step further. They wanted to know, was that material that was being transferred between the different types of bacteria, did it come from RNA? Did it come from protein? Did it come from DNA? They knew about those different types of macromolecules, but they just didn't know which was the one that was responsible for this effect. So what they did in retrospect is a very, very simple experiment, but he treated the samples with enzymes to specifically degrade either proteins or RNA. And we call these a proteinase, the one that's going to degrade protein. The ones that degrade RNA, we call them an RNase. And what he found was that neither of these had any effect. And so this supported his hypothesis that neither protein nor RNA was the genetic material. However, when he treated these samples with DNase, and DNase is the enzyme that digests DNA, he did prevent transformation from occurring. So through this, he concluded that the DNA was the transforming substance. You get rid of it, and you no longer get transformation, therefore it must be the agent that was responsible for this to happen. And that DNA from the dead cells was what was being incorporated into the genome of the living cells. Another important research, some more important research was by Hershey and Chase, and what they did was they used E. coli and they used a virus that will specifically infect it, and we call it a bacteriophage. And this is a virus that's only going to infect any of these phages. They're going to specifically going to infect the bacteria only. And what they did was they were able to demonstrate that DNA, not protein, was the genetic material. The way that they did this was they used some radioisotopes. P32 is used to label DNA. S35 is used to label protein. So by labeling with both of these, they can then follow the radioisotope, and what they found was that DNA entered the bacterial cell during infection and directed the viral reproduction. This was also then being incorporated into the next round of phages. All three of these papers I have placed onto D2L so that you can take a look at them. These are historical papers, and I highly recommend that you go through and just at least read through these different articles. So there's some more evidence that we have DNA is the genetic material, and some of it's direct and some of it's indirect. Some of the indirect evidence that we have is that, well, protein is abundant in the cytoplasm and DNA isn't, right? DNA is going to be found in the nucleus, particularly in eukaryotic cells. We know that DNA is abundant in the nucleus and protein is not. Well, it is present there, but it's just relative concentrations. DNA is much, much more abundant than the protein. We also know that from last week we talked about mitochondria and chloroplasts. They have genetic functions, they have DNA, and these are important parts of these organelles. And they help to direct how they function. And then lastly, DNA is only found where the primary genetic function occurs. And if you do not have DNA, you're not going to have successful cell replication. So if you remember all the way back, I think it was week 1 when we talked and reviewed about mitosis and meiosis, and think about the important steps there for the DNA. If you don't have the DNA present, you're not going to have successful cell replication. So some more indirect evidence. First of all, is that UV light. UV light, RNA and DNA can absorb it at 260 nanometers. Protein can absorb it at 280 nanometers. If you give this different wavelengths of UV light to cells, and you give it to them at 260 nanometers, it is very, very mutagenic for these cells. However, if you give them UV light at 280, there are very few mutagenic effects that can be observed. And so again, this is support that DNA itself is the genetic material. This, however, does not exclude RNA as a potential for having being the genetic material. One of the common ways that DNA and RNA is quantified in cells is by actually calculating what the absorbance is at 260 and then dividing it by the 280. And that'll give you the relative ratio then of the genetic material versus the protein in a particular cell or particular sample. So the best direct evidence that we have comes from recombinant DNA technology. And we'll talk more about this as we move throughout the semester. But basically what you can do is you can take small pieces of DNA from any organism and you can insert it into any other organism. Because the genetic code is universal, because the codons, everything used is universal, this works. Okay, and we actually do use this in terms of harnessing the power of bacteria to make several products for us, and we actually use these for drug targets that we can give to people. So the best example is insulin. You can take the human gene for insulin, you can put it into bacteria, they will grow this insulin product for us, the protein, you can purify it, and then once it's purified, you can then use it for humans. Any diabetic can use this, they can inject it into themselves. Okay. Many other cases. Another really common one that's being used currently is for human growth hormone, and it's performed in a very similar way. Now we do have evidence that RNA can be the genetic material in some viruses. We do not have any evidence that it is a genetic material in any cells, but it can be the genetic material in some viruses. And the key difference between a cell and a virus is that a virus cannot replicate on its own. It needs to have some of the machinery in a cell so that it can successfully replicate. So in these RNA viruses, they have to go through a DNA intermediate, and they have a special enzyme for this called reverse transcriptase. It's an RNA-dependent DNA polymerase enzyme. It takes this RNA and it creates a DNA intermediate. This DNA can then be incorporated into the genome of the host cell. So, whenever the host cell DNA is being transcribed, you can also make copies of this original retrovirus RNA chromosomes. They can then be packaged up, they can be released from the cell, and you have new viruses. Because they have to go through this intermediate process, there's not a lot of proofreading in this process. And so because of that, they tend to mutate very, very rapidly. So some examples of RNA viruses would be HIV, SARS, influenza, West Nile virus. One of the reasons why HIV is so dangerous is because it's constantly mutating, and so it's very difficult to make drugs that will target this virus. The same with influenza. People need to get a flu shot every single year because the flu virus strains are constantly changing.